on Monday we finished talking about RNA, right? We were chatting a little bit, so a couple of things that, like very basic things that I want to remind you. First of all, it's unstable, okay? Second, it's a major molecule that does like messenger functions in the cell. It conveys the info from the DNA through the translation mechanism, through the ribosome, so proteins can be synthesized. Okay, and also serves a bunch of other purposes like transport, ribosomal structure and function, and regulation of messenger RNA stability. And it's kind of a brief overview of those molecules, but as we progress through the various steps of gene expression, we're going to encounter these molecules again and again. So we come back again to the idea of genotype defining a phenotype, right? Basically, we have to understand that genes expressed at the given time will define the processes, biochemical processes that go on in the cell. They will define cellular appearance. One of the best examples is Serratia marcensis, or marcescens. Don't pay attention to my problems with pronunciation. At the temperature slightly higher than room temperature, Serratia is going to produce red pigment. Okay, at 37, it's going to produce white pigment. It's going to not produce any pigment, sorry. Okay, now remember we plated the Serratia when we did transmissibility, and we noticed another thing, that colonies have the property of quorum sensing. When they're crowded, when population is dense, they do not produce any pigment. When population becomes rarefacted, colonies become separated farther apart, they start to produce pigment. Okay, so the environment, actually the conditions, define which genes are going to be expressed, and genes that are expressed will define phenotype. And this is very, very easy to observe in the microbes. Okay, we have to, the same thing happens in mammals, in big animals. Okay, we don't talk about them, but same thing applies to humans, to birds, to cats, I don't know, whatever. Okay, the convenience of microbes is that they're small, cheap, and they replicate fast. So, and nobody's going to ask you, why do you do this to bacteria? However, people in animal care committees in the universities will definitely ask you, why do you want to squish the mouse through the meat grinder? What is the purpose of the experiment? Okay, why do you want to cause so much pain? It's actually a big problem. You have to consider if the animal suffers through the experiment. So you have to minimize the pain. With bacteria, nobody's going to ask you that, right? It's okay. And what about the genome sizes in different organisms? They vary greatly, and as far as I know, we do not have the largest. Human genome is 3 billion nucleotides, which is a lot. But it's not the biggest. As far as I remember, there are some plants that have tens of billions nucleotides. So size of the genome is not strictly related to the complexity of the organism. However, as a general rule, viruses have a smaller genome than bacteria. Bacteria, being prokaryotic, have smaller genomes than fungi or protozoa, okay, eukaryotes generally speaking, and eukaryotes, the unicellular, single-celled eukaryotes, have smaller genomes than multicellular ones. That makes sense? Again, I can give you examples. Pandora virus. Virus so big that it wasn't discovered till about 10 years ago because it was so big. It was unusually big for virus. Nobody actually thought that whatever they saw in the light microscope was a virus. Okay? So Pandora virus has a genome size of 2.5 million nucleotides. The genome size for Mycoplasma is about 500,000. So 5 times difference. You see? But Mycoplasma is the smallest independently living organism. Okay. Now we chat about this, that environmental conditions, especially in the microbes, will determine the gene expression and the phenotype. Now, where are the genes? What are the genes? Well, kind of probably running ahead of the train a little bit. So genes are the functional units of the genome, right? Each gene, we generally say gene encodes a trait, which is not entirely true. It's really hard to give a definition to the gene, okay? A meaningful definition that would reflect something about phenotype and its link to the genotype. But let's so far think about a gene containing that as a unit of a DNA that contains information about a specific polypeptide, a protein, okay? Now these genes, all taken together, compose the entire genome, and genome in the cells exists in the form of chromosomes. Since we are going to focus on bacteria, bacterial chromosomes are circular. Okay? They don't appear very circular in this picture. They don't exist in a shape of the perfect circle. Okay? But if you will stretch them out, you will see that they are circular. Okay? They don't have loose ends. Okay? In eukaryotes, chromosomes are linear. Can you tell me on which stage of the cellular life chromosome would look like that? Will it look like that during meiosis? At one stage, yes, it will. It will look like that during mitosis too, during prophase of mitosis, right? Because generally, I compare chromosomes to a bowl of, like, nucleus with DNA. It's like a bowl of spaghetti with meatballs inside. So that spaghetti, all tangled, is the DNA. DNA in the nucleus that exists in the form of chromatin, like a spaghetti shape, okay? That chromatin is not really assuming any shape. When cell divides, chromatin assumes the shape of the chromosome as we used to know that, okay? And when cell grows, it all looks like that, okay? Now DNA does not exist by itself, doesn't just laying loose in the nucleoid of the bacteria or nucleus of the eukaryotic cell. DNA is bound to the proteins. In the eukaryotes, those proteins are called histones. In bacteria, they're called histone-like proteins or nucleoid-associated proteins, NAP. Any idea what is the function of those histones? Or histone-like proteins in bacteria. So think about this. DNA is wrapped around them, like a thread wrapped around some sort of a coil or something. No, storage, I mean, storage is just, compacting. That's a good idea, but no. It's pretty dense. It's compacted well. Maintain structure. Well, obviously, structure of the DNA that is wrapped around histones and that is unwrapped, those structures are going to be different. So please elaborate on that. What is the, Not really. It's perfectly safe. Now, if DNA that is wrapped around histones, or histone-like proteins, and DNA that is unwrapped have different structure, what else is going to be different? When I say structure, you have to function. Very good. So if structure changes, function changes, right? Now, what is the function of the DNA? It encodes proteins, so proteins get expressed, right? DNA is read by the enzymes. Information in the DNA is read by the enzymes. Does that make sense? Now, which one? Yes. Well, they all encode, but the ones that are unwrapped are the ones that are going to be expressed. Does that make sense? The ones that are going to be read. You can think about histones as the bookshelf. When books are in the shelf, you're not reading them, right? Physically, you just can't. You have to take it from the shelf and open it. You can't open the book when it's on the shelf. I mean, physically, you can't. So, same goes for histones. When DNA is wrapped around histones, it cannot be read. Does that make sense? So, really have to uncoil it so that enzymes that read DNA, such as RNA polymerase, can bind to it and start reading. Does that make sense? No? Does that make sense? So, and actually, all the machinery that regulates that histone binding essentially regulates the gene expression. Does that make sense? Yes. It's a little bit more complicated, but when people say that's why jargon is so bad for science, because when they say unzip it, what does that mean? This, right? When the DNA is, sorry, my hands just can't bend this way, but when DNA is wrapped around histones, it's still double-stranded. When it's uncoiled from histones, unbound from histones, it's still double-stranded. So when RNA polymerase starts to read it, then unzipping happens. Does that make sense? It's the next step. We're going to talk about it. So everything is fine. Now, DNA that we have in the chromosomes, whether it's a bacterial one or eukaryotic one. Have you heard the term junk DNA? You heard. So what do you think it means? DNA that does not encode any proteins. I would be very careful about purpose, because our knowledge about what the function of those fragments is very limited. What is the percentage of junk DNA in the human genome? Well, I mean, give me a wild guess. Come on, people. 75%. Any other? I'll accept 3 guesses. Huh? I'm not accepting any guesses. That was the right guess. 90%. I mean, you were pretty close. People usually hesitate to say something more than 50%. Turns out 90% of the human genome does not encode for any proteins. And similar things are applied to bacteria, to protozoans, to fungi. Any organism, almost every organism, has a lot of DNA that does not encode any proteins. We know functions for some of it. It's usually regulatory, okay? But for a lot of that DNA, we have no clue. And look, nature is very bureaucratic, and most of the processes in nature work according to the principle, don't break things that work, okay? So if there is a fragment of DNA that loses its function, it doesn't mean that this fragment is going to be cut out and thrown away because nobody needs it. It may just stay there for a very long time without any function, okay? So that's what happens. By the way, as far as I know, among the higher animals, the animal with the least percentage of junk DNA is the fugu fish. Have you heard fugu fish? The Japanese fish, if you don't cook. No, they don't cut out. They don't. They cook it in a certain way, and if you cook it in the wrong way, you're going to die. So you have to cook it in a very special way and a special temperature so the toxin will get destroyed. Because if it doesn't get destroyed, then you're going to have respiratory arrest, and you're going to die of suffocation. It's a very exciting experience. I believe so. It's a neurotoxin. Yeah, so pretty much just residual stuff. Yes. And actually, it's the toxin called tetrodotoxin, and it is believed. You've heard the stories about zombies, right? With Walking Dead, everybody knows about zombies. I never watched a single episode, by the way. But it is believed that the legend, the idea of zombies coming from Caribbean, came from those fellas, I don't know how to call them, not the magicians, but those tricksters. They gave people tetrodotoxin at the level that it didn't kill them, made them barely alive, so they were buried because everybody else assumed that they were dead. And then when they got woken up, their brain function was severely affected, and they could perform like basic functions, pretty much like, kill, don't kill. I don't know, things like, it's all kind of half of the hypothesis, half urban legend. But it sounds, at least more scientific key than simply raising from the dead. Now, that's the junk DNA. So your chromosome, not only yours, the bacterial chromosome, consists of a lot of DNA that is dispensable, let's put it this way. Kind of, by the way, to prove my point, there's a guy named Craig Ventnor. He was the first person to completely sequence human genome. It was very exciting. United States government spent like $200 or $300 million in sequencing. Ventnor said, I'm going to do it faster, and he did. He also did it cheaper. So he's a big guy in biotech and stuff. So what they tried to do, they took mycoplasma, 500,000 nucleotides, and they try to find out which genes they can throw away so that the organism can still replicate. They threw away about half of the genes, including junk DNA, genes and junk DNA, and it worked perfectly fine. So it gives you an idea that a lot of DNA is dispensable, actually. DNA can be found not only in a nucleus in the eukaryotes or nucleoid in the prokaryotes. DNA can also be found in mitochondria or chloroplasts in eukaryotic cells. This is so-called extracellular DNA. And we've discussed the sources. It's endosymbiotic organelles, right, that originated from aerobic bacteria, mitochondria, and photosynthesizing bacteria, the chloroplast. Interestingly enough, in prokaryotes, there is also the extrachromosomal DNA, DNA that is not a part of the genome. It exists in the form of a plasmid. Now, plasmid is, again, it's a circular DNA. It's a double-stranded circular DNA. Doesn't assume the perfect shape of a circle. Okay, now why plasmids? They may carry certain genes that are beneficial for microbe. For example, a lot of plasmids carry genes responsible for antibiotic resistance, which is a tremendous problem in healthcare. Because, as we will learn, plasmids can be fairly easily exchanged between the cells. And you can imagine that if you get three cells that are resistant to antibiotic in a population, that resistance will likely spread if you put some selective pressure on the population. That makes sense? It may be a gene that encodes the enzyme that produces some nutrient. And for many pathogenic bacteria, these genes encode virulence factors. Okay? I said toxins here, but it can be any virulence factors. For example, just yesterday I was reading about Bacillus anthracis, the microbe that causes anthrax. It's gonna come up in our discussions on many occasions, not only as a link to 2001 anthrax attacks, but just as a great example of pathogenic bacteria. It carries two plasmids. One plasmid encodes the toxin, which some cellular poison. I don't really remember what it does. It's like I think it's a 3-component toxin, like really complicated. And this plasmid, PXO2, encodes capsule. So it is really well protected against your immune system. That makes sense? So you take those plasmids out of the Bacillus anthracis, and it can be done using molecular biology methods, and it becomes much less pathogenic. You look at the Bacillus cereus, which is a very close relative of anthrax, does not have those two plasmids, and it has different virulence factors. All it causes is the food poisoning. I'm not saying that it's good, but it's much better than anthrax. Okay? Now, DNA. To kind of sum up this idea of the DNA. DNA contains genetic information which either can be expressed. Let's picture it. So DNA can either be expressed, expression of the gene. For some reason I don't like this word, but it's an official term. Or it can be copied, okay, to preserve the genetic information and give it to the next generation of cells. Does that make sense? I want you to understand that those two processes, gene expression and gene copying, are two absolutely not identical things, not related, except that they both involve DNA. Other than that, they are not related. Does that make sense? Good. So, expression of the genes, let's talk a little bit more. Again, you're probably getting tired from that statement. Gene expression defines the phenotype, and in turn, environment defines which genes are expressed. How? DNA has so-called regulatory elements, which means, let's say, concentration of carbohydrates, sugar concentration in the environment changes, okay? That alters biochemistry of a certain protein, okay? It initiates the signaling cascade signaling cascade, and I try to focus on the bacteria, signaling cascade causes changes in protein binding to the DNA, and you have some genes. Oh, come on. Easier, much easier. Sorry, disregard what I just said. Lactose. You have lactose. What is lactose? Sugar. Where do you find it? Milk. So you have lactose suddenly available in the environment. Lactose molecules will bind to a special protein that is called repressor protein, and they will turn the repressor off. Repressor all the time was sitting on the DNA. So when it is turned off, it falls out. DNA is accessible. Genes are transcribed, genes are expressed, okay? These genes encode enzymes that use lactose as the source of energy. Does that make sense? There you go. Environment changes the phenotype immediately. Does that make sense? Good. Now, for a long time, there was a hypothesis of one gene, one enzyme. So if we will try, well, first of all, so you have DNA, right? Then it's being transcribed into the RNA, and RNA is being translated into protein. My analogy, which probably sucks as most of the analogies, does anyone listen to podcasts? Radio shows, okay. When you listen, it's an audio file, but some podcasts provide you with the transcripts, right? So it's all the same words, but just written. So DNA and RNA are both nucleic acids. Slightly different, but nucleic acids. Does that make sense? That's why we call this process a transcription. When you convert RNA into the protein, you go into totally different language. From the language of nucleotides into the language of amino acids. Let's mark it here. Nucleotides, nucleotides, amino acids. So you go from English to French, right? And when you convert from one language to another, it's a translation. So DNA is transcribed into RNA. RNA is translated into the protein. Of course, as we discussed in our example with the cookbook, it is not entire DNA that is being transcribed and translated at every given time. It's a fragments, genes. Genes that, so mechanisms that defines which genes are expressed. This is, whole thing is the gene expression, okay? Expression. Mechanisms that defines are the regulatory elements of different sorts. Now each gene, what does it make? We can present the genes as like fragment of the DNA. So as we decided before, 5' is going to be on the left, okay, 3' is going to be on the right, so it's going to be DNA, right? And then this gene essentially is transcribed into mRNA. Well, actually 3', 5', and it's translated into the protein. So it looks like one gene, one protein. It was original hypothesis of two scientists, Beadle and Tatum. They won the Nobel Prize. I don't remember. I think it was physiology and medicine. So they suggested that one gene encodes one protein, one enzyme, not two enzymes. Which, again, going back to analogy with the cookbook, it means that when you use the recipe for the apple pie, you're not going to make blueberry pie. You're not going to make the beef stew. You're not going to make the Caesar salad. You're going to make apple pie. Just apple pie. Does that make sense? So what they've done, they had several mutants of the fungus called Neurospora. Okay, and they looked at the growth of these different mutants in minimal medium. They defined medium that carries the minimal number of nutrients. Turns out that wild type grew on four different types of conditions: minimal medium, and minimal medium with added amino acids. Okay, and then they looked at three different mutants, and it turned out that one mutant grew only in a medium with either amino acid added. Another one required either citrulline or arginine, and the third mutant specifically required arginine. So here's the thing. There are genes that convert, I think it's serine to ornithine, okay? Genes that encode enzymes that convert ornithine to citrulline, and enzymes that convert citrulline to arginine. So mutant number 3 did not have this specific enzyme. They were able to identify which gene was affected. Does that make sense? So they showed in a very convincing manner that they had 3 distinct mutants with the mutations in the genes that encode a specific enzyme. Okay? So what it means, the gene for enzyme number 3 encoded only enzyme number 3, not enzyme number 2, not enzyme number 1, only enzyme number 3. Does that make sense? So their hypothesis was very simple, and it was extremely elegant. A given gene encodes one specific enzyme. It was a lot of time ago, very long ago, like 60s, maybe a little later. I actually had a privilege to work in when we were in Nebraska. I worked in the Beetle Center. I forgot his first name, but Beetle was working at University of Nebraska at the time, and the huge building that harbored the departments of biochemistry and some other parts of school was named in his honor. It looked like a crematory, actually, like a big crematory. Very depressing. So, as many other hypotheses that were proposed and kind of proven at the time, this hypothesis, I wouldn't say it was wrong. It was incomplete. It turned out that one gene can, not necessarily must, but can encode more than one enzyme. Through the modifications, post-translational modifications, in different conditions, the enzyme that is produced from the gene, there can be different kinds. Of course, going back to the analogy with the cookbook, doesn't mean that when you use the recipe for apple pie, you can do stew if beef stew if you just veer a little bit, off the road. No, it means, when you cook apple pie, come on, substitute apple to pears. It's going to be pear pie. Throw in some blueberries and it's going to be apple blueberry pie. You can modify, but it's still going to be a pie. Same goes for enzymes. They're going to do similar stuff. Very similar stuff. Slightly different, but very similar. One term that I forgot to introduce to you while we talk about this is the term auxotrophic. Auxotrophic means that auxotrophic microbe means the microbe that requires a certain nutrient to grow. Does that make sense? So for instance, this is the auxotrophic microbe that requires what? Ornithine or citrulline or arginine. So it needs at least ornithine. This is auxotrophic microbe that requires arginine. Okay? Often those microbes that are auxotrophic, they have that little signature. For example, you have, let's say, this one, the mutant number 3, would have a writing next to it, arginine minus, which means it does not make arginine. It needs arginine in the medium. That makes sense to you? Sure? Okay, like when people just can't live without chocolate. Okay, so I decided to go with the gene expression first and DNA replication afterwards. So we're going to start with the whole idea of gene expression. Now, most of the things that we're going to cover are going to be about bacteria. We're not going to talk a lot about eukaryotic gene expression. I could say, oh, because it's not important, or because it's not interesting, but I will be completely sincere with you. Because it's freaking complicated, and I don't want to talk about this. I have very bad memories about it when I was in college. I don't want to talk about those bad memories. I mean, we were done with prokaryotic transcription in a day, and then we spent 3 weeks on eukaryotic one. There are like 3 different RNA polymerases, a buttload of regulatory factors. But conceptually, the idea is the same. Okay? We good so far? Now, transcription. You have DNA. In the very basic form, you have DNA. And the result of transcription, RNA. Few questions for you. Question number 1, DNA. In which form, 2 strands or 1 strand, it exists in the chromosome? 2 strands. So, double-stranded. Get used to that acronym DS. DS means double-stranded. Okay? RNA. In which form it normally exists in the cell? Single, coiled, yes, but single or double-stranded? Single. So first, double-stranded DNA, when it is copied, it produces 1 strand of RNA. Based on this information, if you have double-stranded molecule, and you have single-stranded molecule in the end, and that single-stranded molecule in the end is produced based on the complementarity principle, how many strands of the DNA are actually being copied? Just 1, exactly, just 1 strand, okay? This is, sorry, this is important part here. This is called a template strand, okay? You can see it's light blue. I don't know who chose the colors on that picture. Okay, it's light blue. You can see the template strand. The question that should pop up right away, can we just get rid of non-template? It turns out we cannot. It turns out transcription in bacteria, in archaea, or in eukaryotes, transcription always requires double-stranded DNA molecule. Does that make sense? It's really an important, it's a fundamental concept. Steering a little bit away, there are viruses that have single-stranded genome, single-stranded DNA genome. So the first thing they have to do before expressing the genes is to make the DNA double-stranded. So they have to synthesize second strand, and only then they can make RNA. Does that make sense? Yeah, it's going to be used in translation, but so far it's really a fundamental concept. Double-stranded DNA into the single-stranded RNA. And this is the must. Okay? That makes sense? Now it goes from 3' to 5'. Okay? So you see synthesis, first it's 5', 5' end is being synthesized and kind of goes away out of the transcription complex. The transcription machinery in prokaryotes, it's in bacteria, is rather simple. This is the crude representation of the enzyme that is called RNA polymerase. Okay, so polymerase means it produces a polymer, which is called RNA. Does that make sense so far? Now, this structure doesn't really, I mean, it kind of shows you what's going on, but actually, if you would be able to take this complex and start rotating it to look how it looks like, it vaguely reminds this. Okay, incomplete feast. So when DNA goes through that opening in the feast, and RNA kind of hangs out of here. Does that make sense? So pretty much the enzyme slides down the DNA. That makes sense? I was studying this process when I was in undergrad. I was doing the project on this. So RNA polymerase of bacteria bind to the fragment of the promoter sequence. Now, what is promoter? Any idea? What would you think the word promoter means? Yeah, exactly. It's like, like runners, before they run like 100 meters, they have a good stretch where they can like walk to the starting line. Okay, starting line is not just right from the fence, so it's not like this. Okay, they have some space behind them. That's a promoter. So RNA polymerase binds to the promoter first, and then it starts transcription from the starting site. So you can see it's kind of shown here. Well, barely shown here, the promoter site, but this is, I think, better. So you see this is the +1 position. So it's the first nucleotide that is being read. Does that make sense to you? First nucleotide that is being read. So RNA polymerase binds to the promoter, and it recognizes sequences in the -35 and -10 area. Actually, there are special like sequences of nucleotides. Like -10, you just happen to know it's TA. And you can see, if you would look at the promoter region close to this initiation of transcription, the transcription start site, it is AT reach. Why? Huh? Why? AT reach. A lot of adenine and thymine residues around the starting site. Huh? What about AT pairs? The number of hydrogen bonds between A and T. Huh? That's weaker. So what should happen at the promoter site? Unwrap, right? You start, well, not you, but RNA polymerase starts to unzip. You just mentioned unzipping, right? So it starts to unzip. Remember, we have one strand copied, so we have to get them separated. That's why a lot of ATs, yes. No, wait. those are two anti-parallel strands of DNA. Okay? In order to start the transcription, you have to unzip them, separate them. The two strands of the DNA molecule. Because you're going to copy only one. Right? Only one. That's the point. So, that is shown here. It unwinds at the initiation site. I don't know if you can see that unwound DNA. You can see it here. By the way, that part of the DNA which is unzipped, it is called transcription bubble. And that bubble slides together with RNA polymerase down the DNA molecule. Does that make sense? Not really? No? Okay, so it's like this goes through them. So behind part is double-stranded again. The part in front is double-stranded again. Okay? But the part here is unzipped. Now, a little crash course in terminology. This is called upstream. Anything before the initiation site is upstream, well, this is downstream. Okay? Now, RNA polymerase, as anybody else, isn't perfect. Which means it starts transcribing and then falls off. Starts transcribing and then falls off. This is called abortive initiation. This falling off. So it produces short, really short fragments of RNA, like 10 nucleotides or something. And then they're going to get killed. But once the complex between the RNA polymerase and the DNA is stable, the bubble is stable, elongation starts. Okay? So here's the elongation. Now, we've talked about the first stage, initiation, which consists of binding of the enzyme to the promoter region, unzipping DNA, and that little abortive stage. And then we talked about, and then initiation goes into the second stage, the elongation stage. Okay? Actually, it's pretty boring what happens afterwards. Termination. Enzyme stops moving forward because of specific sequences. The terminating sequence just falls off. Does that make sense? And then you've got your RNA transcript in the end. Okay, that little green stuff at the bottom that you can barely see. That I can barely see. Now a little, again, wearing to the side, splicing. So I will even write this statement so you have it in writing there, you have it in writing in your notebooks, and you have it on the video. Okay. In fact, archaea do not splice as well. Okay? So only two, so eukaryotes splice, yes, all of them. And interestingly enough, viruses splice too. Now what is splicing? Opposite. It's actually cutting out the crap. Remember we talked about junk DNA. This junk DNA is not distributed in the genome like, oh, we have 10% of the meaningful stuff and then 90% of crap. This junk DNA, the rubbish, is dispersed. in a genome. So in a given gene, right here, you're going to have meaningful coding fragments that are called exons. Make sense? Exons in the gene are separated by non-coding fragments, introns. So when in eukaryote the gene is transcribed, both exons and introns are transcribed into mRNA. Does that make sense to you? But you don't need rubbish. You don't need the junk sequences. Special, like huge protein complexes that are called spliceosomes cut out the introns, stitching exons back together. Does that make sense? Look, it's like editing. It's editing of the video. Okay? You have like an hour and a half long video of something, but there are fragments that are not really exciting. So you upload it on the computer, find those fragments, and splice them out. And then stitch the meaningful fragments back. Does that make sense? Now, probably the most important consequence of the splicing is the concept of alternative splicing. If you have a gene with multiple exons, sometimes it's 2, sometimes it's 6, okay? Here, in this example, there are 5 exons. If you combine those exons in the different ways, all 5, or you, for some reason, the third one is excluded, or the fourth one, the protein, in each case, the protein product is going to be different. Does that make sense to you? You've pretty much taken out parts, right? Again, kind of, we do splicing I don't know about you, but when I cook, I pretty much do splicing every time I upload the recipe from the internet. Because sometimes recipe requires for some ridiculous things that I don't have in the house, so I just throw it away and hope for the best. Yes?
So this is like the whole idea of gene expression.
We're getting through this. So expression will be done when we will talk about translation. This is the step of the gene expression that is present only in eukaryotes, not in bacteria, not in archaea. Okay? And we will summarize it. We look at the smaller parts, we'll learn it bit by bit, and then we're going to have the overview, absolutely. Okay? This is the idea. Do you get the idea of splicing? Okay? Yes? Splicing is between the transcription and translation. So yeah, you have to get transcription first, and then splicing, and then translation. Okay? So bacteria, they skip this step. Okay? They don't have splicing. The genes do not have exons. They have different distribution of the junk DNA. Okay? Good? I want to apologize, but I don't want to apologize. I know, this is the very text-heavy slide. The reason why I make it text-heavy, it's not for me to read it. It's more for you to get the reference point, okay? Does that make sense? So that's why I tell you, you don't need a book. So let's talk about the translation and the genetic code. First, let's get out the translation concept. Let's get it out of the way. Translation is the making a protein based on the information that mRNA contains. Does that make sense to you? mRNA carries certain message, information that was once a gene, and then special organelles, polyribosomes, they translate that information from the mRNA into the protein sequence. All right? That makes sense? Now, what is the ribosome? Is it membrane-bound organelle or not? Is it surrounded by membrane or not? No, it is not. Ribosome is a structure that consists of RNA and DNA. So prokaryotic ribosomes actually consist of two subunits, the smaller 30S and the bigger 50S. Structure of eukaryotic ribosome is different, but basically it's also two subunits. Okay? What pretty much happens is you have a ribosome and the mRNA bound to the small subunit. All right? Why we call them polyribosomes? Huh? Yeah, there are many of them sitting on the mRNA molecule. Does that make sense? So far, let's see which big conceptual difference we learned between pro and eukaryotes. First one, prokaryotes do not have splicing. That's one. Second, in prokaryotes, transcription, mRNA synthesis, and translation are continuous. Now, let me explain what I mean by that. Huh? Yeah, so in prokaryotes, transcription and translation make continuous process. I'm going to explain what I mean by that. Look at this specific illustration here. And this one actually. Okay. So this is RNA polymerase, which produces mRNA in the process of transcription. Once mRNA gets out of the complex between DNA and RNA polymerase, it immediately gets translated. Oh, horrible, horrible mistake, I didn't notice. It's not DNA, it's a protein. Idiots. Well, I should be more better to them, okay? It's really a tough job to find such small mistakes, okay? So you see, mRNA gets out and is immediately translated. Does that make sense? Right away. It's like going back to the cookbook analogy. You're going to get tired with that analogy. You have a really, really long recipe, and your mom stands in the living room and yells at you. She doesn't even copy the recipe. She yells at you, hey, get 2 cups of, I don't know, whatever, 2 cups of wheat flour, okay? And you take, well, what's next? Do this, and you do this. You don't know what you're going to do next, okay? So this ribosomes, they don't really know what they're going to translate in, I don't know, a few milliseconds, because it's not even synthesized yet. You can see it better here, okay? So you have RNA polymerase on the DNA, which produces the violet or purple, whatever you want to call it, mRNA. And ribosomes immediately start to bind to that mRNA, producing proteins, which looks like a bunch of beads here. Thingies, good. I will explain what each of the thingies are. Good that you told me because, you know. So blue stuff, blue line, that's the DNA. That's the DNA. Are you talking about this one? You can barely see it. Can you see it? No? I'm going to make it like really, really blue, okay? That's DNA. We good? Those little beige things. I don't know if you can see them. I mean, you can barely see them. I know that they are there. That's why I can label them. Those beige things that I just circled black are RNA polymerase, the molecule, the enzyme that makes RNA. The purple lines are the molecules of mRNA. So these actually are not steps. That's kind of a snapshot taken at a given moment. So I have 4 transcripts, 4 mRNA molecules in the process of synthesis. That makes sense? Those other beige things that abound to that purple line are ribosomes. Okay? And those bead-like things are proteins. Okay, these are ribosomes. Does that make sense? Not just proteins, growing proteins. So so you see, transcription still goes. mRNA is not finished yet. It's not done. But translation already occurs. Now, all of this continuous process can happen only in the prokaryotes. Why? It can't happen in eukaryotes. This continuous process of transcription and translation can happen only in prokaryotes, but not in eukaryotes. What's not a problem? What? No, it's not about energy. Think about the fundamental difference between pro and eukaryotes. Okay, nucleus. So which eukaryotes have it? Prokaryotes don't. Wait a minute. We're going to get into like real mechanics under it. Nucleus. Does it have a physical boundary? What is the boundary? A membrane. In the eukaryotes, transcription, where does it happen? Transcription. In the nucleus. Translation. Cytoplasm or endoplasmic reticulum. So these two processes, transcription and translation, are physically separated by the membrane. mRNA that is made in a nucleus first have to leave it. There is no way these processes can happen at the same time. Absolutely no way. It's like you make one part in Illinois and then ship it to Michigan, okay? You just can't work on the part while it's in the making. Does that make sense? In prokaryotes, there is no membrane. DNA is, in a second, DNA is transcribed, and that unfinished transcript immediately translated. Yes? Yeah. Ribosomes in eukaryotes, it's in the endoplasmic reticulum, in the cytoplasm, it's everywhere. But the point here is nuclear membrane. Okay? Does that make sense? Now, going back to the ribosomes, we're going to chat about ribosomes a little bit, and before we get to the genetic code, we can take a break. Ribosomes. Well, we kind of talked about the difference. They are different, right? Eukaryotic and prokaryotic are different. They're structurally different, which means we can selectively inhibit one or another. Okay. Interestingly, mitochondria and chloroplasts also contain ribosomes, and they carry out their own process of transcription and translation. And the ribosomes that are found there in the mitochondria and chloroplasts are 70S, like prokaryotic. Do I need to explain why? Not really, right? I mean, mitochondria and chloroplasts are former bacteria, so they just kept those. So even ribosomes in the mitochondria and chloroplasts are different from the ribosomes of the eukaryotic cell that mitochondria and chloroplasts live in. That is another point towards the symbiotic origin of those two guys, right? Now, before we go to the break, one thing that I wanted to quickly mention. We just talked about this, about this difference, okay? I'm going to put the note here. Selective inhibition. We can selectively inhibit the function of the bacterial ribosome without affecting the eukaryotic, the human one. Does that make sense? I can tell you more. Remember I told you that I'm not willing to tell you about eukaryotic transcription because it's big and I hate it. Okay? Well, obviously, prokaryotic RNA polymerase and eukaryotic RNA polymerase are very different, which means we can selectively inhibit this dude right here. Guess what? There's a drug that does exactly this. I hope you never heard of it. I hope you never will. The drug is called rifampin. Okay, have you heard? Good, good. I really like that you either don't know it or you know it but don't know what it does. It's a drug against TB, one of the best drugs against tuberculosis. Okay, it is fairly selective inhibitor of RNA polymerase. And if we'll talk about inhibitors of ribosomes, the translation in the microbes, tetracycline, heard? You've heard about it? Erythromycin, azithromycin, Zitromax, antibiotics, neomycin, the kanamycin and other streptomycin. You probably, I don't know if you've heard linezolid. It's kind of more specific, again used for TB treatment. I'm trying to think what else we have. Not ciprofloxacin. I forgot one type. I don't remember it from the top of my head. Okay, but those four that I gave you, like, it's a major types. Aminoglycosides, macrolides, oxazolidinones. Those are classes of antibiotics that inhibit specifically the bacterial ribosome. Okay, so it's pretty cool. The reason, what is the reason we discover more and more new antibiotics? Yes, resistance. And the sad truth about antibiotic resistance and antibiotic discovery, that in the last 40 years, there was not a single new class of antibiotics discovered, which means like cephalosporins. I don't remember like ceftriaxone and other drugs, there are 5 generations of cephalosporin antibiotics. So we discover one drug, and then we start making it better. But mechanistically, it's still the same drug. We discover one drug like clarithromycin. Well, it wasn't clarithromycin, it was something else. I don't remember the initial. I think it was lincomycin. And they start to make it better. Okay, so it's the arms race. And microbes always going to outrun us. Okay, let's take a break. And when we come back,